Hi, welcome back. Today I'm going to be discussing part two of this kind of mini series going into depth on the effect that fats have on cancer progression, and things of that nature. So if you haven't checked out the first episode or the first part of this series, then I would certainly recommend that you go over to that video, which I'll link for you, because it goes into some serious depth into the cell mechanisms of cancer and how fat can have a specific impact on those cell mechanisms and things of that nature. Now, even though I ended up talking for, I think, like 45 minutes or an hour in the last episode, rest assured, I mean, this is just a drop in the bucket of the information that scientists and researchers know. But I wanted to, and I kind of tease this as I often do, at the end of that episode, I mentioned that I would be discussing some therapies as well that are related to fat-specific cancers or fat-reliant cancers. So that's what this episode is on. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine, and I work in a cell biology laboratory. So, and the information that I'm going to be talking to you about, certainly I'll be filling in the details, but the bulk of the information is actually going to be coming from a scientific review on the literature on cancer research. So, with that introduction out of the way, let's go ahead and jump into this. Of course, I'm going to have graphics for you as we go through it, as I tend to do. And the question that we're going to be examining is what therapies exist against fat-focused cancers. And I did mention that I'd be discussing nutrition therapies as well, so I'll be adding that near the end. But first, I want to discuss some more of the drug or pharmaceutical therapies that are present, and also offer a problem that I think that a lot of people don't fully understand why we can't solve the issue of cancer. I'm going to present that problem to you so that you understand. And all this information comes, well, not all of it, but a lot of the information that I'll be going over comes from the British Journal of Cancer, this particular review called Reprogramming of Fatty Acid Metabolism in Cancer, fatty acid being fat molecules. And this is linked in case you would like to look over my notes or look over the paper itself. All right, so a bit of background to recap just a few snippets, a few highlights from the previous episode. We discussed a series of different ways that fatty acid enzymes or fat molecule enzymes, fat-related enzymes within your cells have an impact on cell growth, cancer metastasis. We talked about cholesterol, we talked about saturated fats, we talked about unsaturated fats, and then we talked about these particular enzymes like the one that I have right in front of you right now, which is called fatty acid synthase. And I had this diagram, it was slightly different from this one, but ultimately it was a situation where a cancer, so this cancer right here, would experience cell division and cell growth if a ligand or a hormone or some sort of protein or some sort of particle, molecule binds to this receptor on the outside of the cell. When that happens, then it leads to a triggering event of a series of different molecular changes that I've completely glossed over by this one arrow. But in reality, there's a ton of different changes that happen within the cell once this ligand binds to the extracellular section of the cell of this receptor. Now, what was intriguing is that with some cancers, and this is really important that you understand this, that I'm saying some cancers, so I don't want you to think that this is ubiquitous to all cancers or anything like that, but some cancers have this heavy reliance on this enzyme known as fatty acid synthase. What it does is, as it's shown here, is that it takes an acetyl-CoA molecule, and this is going to be important because I'm going to be discussing this in more depth later, but it takes an acetyl-CoA molecule and generates a saturated fat from that. And somehow, although we don't really understand the mechanism, or at least it wasn't described in the review, somehow the presence of this fatty acid synthase and the greater activity of this fatty acid synthase allows this whole action to occur, as in you would normally have binding of the ligand to the receptor and then changes intracellularly, but this seems to supercharge the effect. And what researchers have tried to do then is try to knock out this enzyme and see what effect that has on cancer cell growth and division. And what they found, again, this is just a recap, but what they found is if you knock this out, that the cells not only no longer grow, no longer divide into new cells, but they actually end up dying. So clearly at this point, and somebody had actually commented, well, then why can't we attack this enzyme? Well, done. We figured it out. Solution. There's no more cancer to be had. Unfortunately, it's not that easy. And the reason for that is because if we use a FASN inhibitor, which is the FASN, so it's just a shortened version of the fatty acid synthase. So the fatty acid synthase inhibitor, so we use that in cell culture models, so just cells, so cancer cells in a dish with the liquid that's keeping them alive so that we can study them. This is the type of research that I do. In my master's, I did a combination of that with some animal stuff, and then I did some stuff on the side with humans as well. But for the current research that I'm doing, it's very much focused on cell culture stuff, which is what we're talking about here. So when they use this inhibitor on the cell culture, that's exactly what they found. That's how they got this result up here of this change. So knocking this out or inhibiting it led to loss of cell growth and division, and therefore cell death. So you can see that very clearly when they could see it very clearly when they were looking at these cell culture plates. However, once they wanted to move up, let's say they wanted to look at animal studies, or they started using this in humans, they found that it led to very severe toxicity, or maybe I wouldn't necessarily call it toxicity in this situation, but there were consequences. There were severe side effects. And two of those side effects, two of the major ones, were severe weight loss and anorexia. It's not like toxicity in terms of like destruction of their liver or something along those lines. It's not to that degree, but it still led to severe weight loss and anorexia, which is clearly a problem because if people aren't eating, then they're going to, of course, lose a lot of weight. But then, of course, you're messing with an enzyme that has an impact on the production of new fat, which it makes saturated fat, so it's not necessary to have that. So that's probably why they were able to use it in the first place. But even so, even though the mechanism that I don't know the mechanism for why inhibiting fatty acid synthase led to severe weight loss and anorexia, ultimately this is not a sustainable model. So let me explain to you how that drug exactly works. So the drug would work through an offshoot system. So here we have our same diagram. We've got our receptor, it's bound by the ligand activity, and then you get the activation through a fatty acid synthase that somehow gets involved into this, leading to cell growth and cell division. But another fate of fats is that they can be oxidized, which means that they can be used for metabolism. They can be used for energy production in some capacity. So a saturated fat will be outside. So I guess I'll back this up real quick. This is on the cell membrane, right? So this is right here, outside of the cell versus inside of the cell. So that's what we're looking at here. Here, we're looking at things in the mitochondrion. So this is on the outer membrane of the mitochondria, and all of this was already explained as well in the previous episode. But this enzyme known as CPT1, or carnitine palmitoyl transferase, is one of two proteins, technically three proteins, but two proteins by this name of CPT1, and then there's another one called CPT2. What they do is they allow fats from inside of the cell, not outside of the cell, but inside of the cell into the mitochondrion. So there's like three different compartments: outside of the cell, inside the cell, and then inside the mitochondrion. So this saturated fat, or this fat in general, would then enter the mitochondrion through, or be allowed to enter the mitochondrion because of this CPT1 enzyme. Okay, so why am I telling you all this? Because this enzyme is actually antithetic to the fatty acid synthase. What do I mean by that? Is that if you're burning fat, you're not going to be producing fat normally. There are certain instances with certain cell types, like fat cells, where the rules get broken a little bit. But overall, if you're breaking down fat to produce energy, typically you're not going to be generating fat. So there's some crosstalk between these, and usually it goes the opposite direction, as in you get an increase in particular precursor molecules that allow fatty acid synthase to work. And what they do, they not only allow fatty acid synthase to work, they actually also inhibit CPT1. However, there's also reverse actions. So if you have the uptake of fat into the mitochondria, you get more oxidized. And we're not so much interested in the fact that these fats are oxidized, that they're used for energy, that they're broken down, utilized for energy. We're more so interested in the fact that this is more active. CPT1 is more active. Therefore, there's an inhibitory effect on fatty acid synthase. And that's exactly what these drugs did. So what they did is they ended up hyperactivating or super activating, or activating more than normal, this CPT1 enzyme, which then led to an inhibition of the fatty acid synthase. Now, one of the problems with that, however, is that CPT1 is a really crucial enzyme for so many different tissues. So there's going to be a lot of widespread effects that can affect the health of the entire body. I mean, we're talking like muscle cells, we're talking about kidneys, just any system except for certain ones like the red blood cells. Neurons tend to have fatty acids metabolized by other cells known as astrocytes. But for the most part, you have a lot of reliance on this enzyme. So even if you're hyperactivating it, that can, of course, lead to widespread effects. Now, widespread effects in terms of affecting the cancer, that's great. You're inhibiting fatty acid synthase. So these cancers that need this fatty acid synthase to function, they'll be strongly hurt by this hyperactivation of CPT1. Great. However, suddenly you have to think that if you're increasing the activity of this, you're also massively intaking tons of fat into the cells and burning it off. Now, that might seem great in terms of a weight loss drug or something along those lines, but you don't want things to go too far into one direction, towards burning off fat, because eventually you end up dying. You can't do that indefinitely. So, and of course, pharmaceuticals getting in the way of that can lead to a series of negative effects beyond just anorexia, the lack of wanting to eat, and weight loss. So overall, this was a clever idea, but it wasn't going to work because of the widespread effects that it had on the body. Okay, great. So that was one therapy that they tried. That was an old version, quote unquote, old version of this therapy. So another thing that they decided to do was, another way of attacking this was to attack a less prominent enzyme that is a precursor enzyme to this fatty acid synthase. So again, this is the same thing. The acetyl-CoA molecule is then converted into a saturated fat. Now, downstream of that, or maybe I suppose I should say upstream of that are other enzymes that convert other molecules into precursor molecules like acetyl-CoA. One of those is this enzyme known as ATP citrate lyase, or ACL. It's sometimes abbreviated ACLY. And it takes a citrate molecule and will convert that to an acetyl-CoA and will then allow fatty acid synthase to produce saturated fat. So if we inhibit this enzyme, which is less crucial than CPT1, then we don't have as many or nearly the same amount of consequences of side effects that we do when we inhibit CPT1. So that's what they've done. They've found ways of inhibiting this ACL enzyme, and it turns out that yes, that it actually does help. It affects the cancer cells disproportionately more than it affects the physiological tissues, the healthy tissues. Are there still going to be side effects? Yeah, probably, but they're going to be far less severe than the ones that we discussed with the previous version of the same. So what I'm pointing at here is that there are a number of different ways of attacking this fatty acid synthase enzyme, and these are two of them. Okay, so that's one therapy. Another therapy that researchers have tried is to, and you've probably heard of this enzyme before, is AMPK, AMP kinase. And AMPK has so many different functions, so let's be real, but I'm going to focus on just a few of them. So one thing is, if AMPK is activated, it typically signifies a low energy state. Now, there's a number of different activators which I will show you, and they will be rather overwhelming, but just for exaggeration's sake and just for interest's sake, I'll show you a number of different activators of AMPK, and I'll discuss a nutritional method that may activate AMPK. But hold on to your horses. Let's just take one step at a time. So I've never understood that expression, hold on to your horses. Hold your horses. I guess because you don't want them to run away? Maybe that's it? If you know, please let me know. Anyway, AMPK activation has two major functions that we're going to discuss here, and I'll add one more at the end. AMPK can lower the activity of, or desensitize the activity of these different receptors, specifically androgen receptors. So receptors that would bind like testosterone, which could be found outside, but typically are also found inside the cell. But really the point is that you have these receptors like the ones that I mentioned earlier, where you have ligand that binds the receptor, and then you have cell growth and cell division. Well, AMPK can have an effect on that receptor, decreasing its effectiveness. So increased activity of the AMPK decreases the activity of particular receptors that would generally lead to more cancer cell growth and division. Another activity of AMPK is to stimulate CPT1, which we've already gone over. CPT1 then would inhibit fatty acid synthase. Now, I can imagine that one of the questions you would have then in that situation is, well, we just went over that and there were some negative effects of that. Well, that's true, but in this situation, the researchers may be, and this is speculation on my part, the researchers may be modulating within a physiological level. So they're not they're not supercharging CPT1 activity. They're just increasing CPT1 activity within a normal level, and that may lead to some inhibition of the fatty acid synthase, which would still have more kind of increasing negative effect on cancer cells, and although it would still have a negative effect on other cells as well, it would be less detrimental than it would be for the cancer cells, because it's just multiplied in the cancer cells because they rely on this enzyme so much more than the physiological cells. Okay, so AMPK is another method, so using drugs to activate AMPK so that you get both of these effects. All right, so that's all well and good, right? So I'm going to show you three different things. So one of them is that you have AMPKs activated through phosphorylation, just like many other proteins are, or many other enzymes, protein, enzyme being the same thing. And one of the main drivers of activation is through the activation of an upstream molecule, enzyme, protein, known as LKB. So LKB will phosphorylate AMPK, which will increase AMPK activation and therefore lead to the effects that we just talked about. However, in a lot of cancers, what happens is that LKB is inhibited because it is mutated. So the protein, the amino acid sequences that make up LKB are not in their normal generic sense. They're not there as they normally would be. They are in some way defunct. They're somehow dysregulated. Like you're missing particular parts. It's like a car that is missing one of its hoses that it needs for the coolant system, or it's missing it's missing all of its spark plugs, like things like that, like important aspects that it needs for it to actually function. So this is a mutated enzyme, and therefore you don't get activation of AMPK. That's why this phosphorylation isn't present. So what researchers have thought is, well, in these cancer cells that may be reliant on fat, so two conditions: one, they're more heavily reliant on fat, and secondly, they also have mutations in LKB. Then in that situation, then why don't we find alternative activation states? Okay, fair enough. And one of those, I'll go ahead and throw this out there, one of those is through a drug known as, and you may have heard of it, is metformin. So metformin lightly activates AMPK. Now, one of the drawbacks of the activation of AMPK is normally it isn't sufficient to just activate it through one pathway. You typically have to activate it through a number of pathways. There's certain pathways that are more potent than others, but definitely if you have a mutated LKB, you need a strong activator of AMPK. So a light activation through something like metformin might work to a degree, but it may not be the best way. But one of the positives, of course, of metformin from a pharmaceutical standpoint is that it is, for lack of better words, kind of a miracle drug in many ways. I'm sure it's not perfect in every way, but it helps in diabetes, it helps in cancer, it helps in a lot of different conditions. And that's because of its mechanism of action, which, if you're not familiar with its mechanism of action, I have a video that details all the cellular components of what changes from taking metformin. So I definitely recommend that video. However, it is a really complex video. It's only like a 6-minute video, I think. It's pretty short, but I think at the very beginning I mentioned this is the most complex video I have ever released. So hopefully I do justice to explain it well enough. So you've been forewarned. It is at this point now one of my more complex videos. So you've been forewarned if you want to know how metformin functions, its actual mechanism of action, where I do discuss how it affects AMPK. All right, I digress, however. But there are other drugs as well that researchers and doctors can then end up using for an alternative activation of AMPK. Let's look at some of that alternative activation. Let me go ahead and get rid of this. So AMPK, clearly this doesn't look like my version of AMPK that I have right here, but it's the same thing. So it's just these different sections of the AMPK, alpha, beta, and gamma sections of AMPK. Now, this phosphorylation site that I was talking about right here is the same phosphorylation site that's mentioned right here. So it's a threonine, the 172nd threonine, which is an amino acid, on the AMPK enzyme. Now, this is really overwhelming. That was kind of my point. But here we see that metformin is one of the regulators of AMPK through LKB1, which, of course, if it's mutated, it may not work, but also through the regulation of ATP and AMP levels. So if AMP levels increase and ATP levels decrease, you get more activation of AMPK. So that's another mechanism. Resveratrol. And then a series of other drugs as well. So like ICAR, and I'm not even going to try to pronounce that. Well, yes, I am. Thiazolidinide. No, I'm not. So statins also affect LKB1, but clearly if you have a cancer that, again, has mutated LKB1, depending on the mutation, that is also important, could still work through LKB1. And then you've got KMK. So the point is that there's a bunch of different regulators of AMPK. And yes, we're going to talk about diet and exercise. Exercise being a big one, although it's rather temporary, but dietary restriction can be a pretty potent one as well. All right, so these are all the regulators of AMPK, and then AMPK is you can see all the different areas that it has a downstream effect, like it activates or inactivates all these different other enzymes, like it inactivates mTOR, it inactivates I think acetyl-CoA, carboxy, something along those lines. It activates CD36, which is a fatty acid transporter. So there's pyruvate kinase, GLUT4. I mean, there's PGC1 alpha. There's all these different regulators of AMPK. That's not important. What's more important is these up here in terms of trying to activate AMPK. Okay, so that's a series of different drug conditions wherein you can use different drugs to target these different things. But the big problem that I mentioned, or maybe I didn't fully mention it, is that how do you specifically target cancer cells? And the point is, we can't. That's the major problem. However, what we can do is, and I'm not done with the discussion, I'm going to go into nutrition next. But what we can do is try to focus our attention so that we affect all the cells, but we specifically target molecules, enzymes that are cancer cells are extremely reliant on those molecules, more so than the physiological tissues. Hopefully you're with me on that. But overall, no. If you could figure out a way to specifically target just cancer cells, that would be a game over. There would be so many different mechanisms that we could attack that would immediately kill cells. I mean, we know how to kill cells. I know how to kill cells. I can kill cancer cells any day I want, no problem. But once they're inside of a body, that's a whole different story, because you're trying to distinguish the cancer cells from the physiological tissue. And you can do that through imaging and stuff like that. But in terms of targeting it to actually damage it, you're going to end up damaging the entire system, unfortunately. Okay, so now let's go into a little bit on nutrition. So 2 different nutrition principles that I wanted to discuss. Let me quick check my notes to see if I've missed anything on anything else. Oh, I should mention, yes, I'm glad I checked my notes. This is going to be important, so I'm glad I checked my notes. AMPK stimulation, according to this review, does not universally damage or hurt cancers. Sometimes cancers will actually be activated, as in, to clarify, you already have cancer. It's not like you activate AMPK and suddenly you have cancer. That's not how it works. You already have cancer. You activate AMPK and the cancer becomes more aggressive, as in it migrates to different areas of the body. So, let me go full screen for this. So it is important to characterize the cancer that a person has before making any sort of changes to your diet or trying to target different things. So you would need to get some sort of like biopsy and then do a genome scan or test things out. Like it's an expensive and long process, but if you can figure that out, you can use nutrition weapons on your side to then target that particular cancer. Target within the confines of what I discussed as targeting, because you're still going to be affecting the rest of your body. Okay, so AMPK, point is that AMPK activation is not always a positive thing. Sometimes it can lead to more aggressive cancer, and in other instances, it can be really potent against cancer. So great. All right, low carbohydrate diet. So a low carbohydrate diet will increase the expression, or generally at least the activation, the use of CPT1, which is what we mentioned earlier. Another thing it will do is decrease the activation of this fatty acid synthase. So it has, and the reason for that, I guess I'll explain that. So the reason for that is not because of the low carbohydrates necessarily, although that's part of it, but actually because of the increase in fat consumption. So it's the same thing, right? If you were to consume a high carbohydrate diet, your cells start to burn more carbohydrates. That's the metabolic flexibility that people sometimes discuss, is the ability for your cells to switch from carbohydrate utilization or oxidation to using fat for utilization, oxidation, energy production. So a low carbohydrate diet lowers the carbohydrates, obviously, and it increases the fat consumption, thereby forcing the cells, as many cells as can switch to using more fat for oxidation. As a result, this leads to greater utilization of CPT1 because CPT1 allows those fats into the mitochondrion to be oxidized. Now, as a result, you don't need to be generating more fats. You don't need to be generating more saturated fat if you're already consuming fats. So as a result, you get reduced activation of fatty acid synthase as it's downregulated because the body simply doesn't need more of it. So the cancers that rely a lot more on this will then be disproportionately affected by this loss of fatty acid synthase. The second thing that I promised I would discuss is the activation of AMPK, which I'll before I continue with this, I'd like to say that at the end of this episode, I'm going to be releasing a video that you should watch if you're interested in what I'm about to talk about, AMPK, and if you're interested in fasting. Then I would watch the next video because once it releases, because I ran across a paper looking at AMPK activation levels in fasting, and there's particular population of people, that's quite a phrase, particular population of people, a lot of P's there, that may not get as much of a benefit from fasting on their AMPK levels. If you're interested in that, then I would highly recommend that video, which will be linked for you as well. Okay, with that said, with that tease out there, let me go ahead and discuss this. So fasting generally increases the activation of AMPK. Now AMPK, although I don't have it labeled here, and we did see that earlier, it inhibits the fatty acid synthase because of the downstream molecules that it affects that we saw in that crazy diagram, like the effect on ACC, which is one of the upstream molecules of fatty acid synthase, but also through what we talked about is the increase in the expression or the activation of CPT1, which we've already talked about multiple times. So fasting does increase AMPK activation, which inhibits fatty acid synthase. Again, assuming that the cancer is reliant on this mechanism. But of course, it's not like AMPK just has this effect. It has an effect in many, many different ways on these fat molecule or fat synthesis or fat related enzymes. Now, the second thing that it does is it increases autophagy. I know everybody goes crazy. Autophagy, my God, it's autophagy. And then take pictures like it's going down the runway. The autophagy is really beneficial in certain cancers, fighting against certain cancers. There's actually certain types of cell death that occur through autophagy. However, I remember somebody commented a number of weeks ago at this point, and it was a really great comment where they were discussing autophagy, and they were saying they'd run across some literature or some people that had talked about how autophagy isn't always beneficial for cancer. And that is absolutely true. That again, you would have to characterize the cancer to figure out if it is susceptible to more autophagy. If that's the case, then increasing AMPK levels would be fantastic. However, if you have a cancer that takes advantage of autophagy, and the way that it might do that, maybe I'll do a separate video on this, but the way that it would do that is by increasing, so autophagy would increase, which is essentially what's known as proteolysis. You're breaking down proteins, you're breaking down parts of the cell, and then you spit out the components of those, and then you can recycle those. So if the cancer cell is using that to its advantage, it's not trying to inhibit autophagy, it's actually trying to increase its autophagy, then it can recycle components of itself as a result. So that's one advantage of having autophagy in cancer cells. However, like I said, certain cancers try to diminish autophagy as much as possible. And in those situations, increasing AMPK would be fantastic, specifically and one of those ways that you can do that is fasting, as I'll show you in the next video. All right, to double check that I haven't missed anything, all right, let me go ahead and talk about one more thing. They also mentioned omega fat, so this is from the review. They also mentioned omega fat consumption, so I'm talking like omega-3s, omega-6s. And they mentioned that, obviously, this is pretty well known at this point, although I'm sure there's a lot of nuance that needs to be added. Omega-6 fatty acids, which are unsaturated fats at the 6th carbon site, all you need to know is omega-6s, are a lot more common in the Western diet, in general diet. And those omega-6s tend to be more pro-inflammatory. Okay. And pro-inflammatory isn't always a negative, but when you're talking about a chronic state of omega-6 fatty acids, being pro-inflammatory is a negative state. And these omega-6s, what they can also do in a specific cancer-related context, is they can be bound by these COX enzymes. Those are enzymes that I mentioned in the last episode. And those COX enzymes will take fats and convert them to prostaglandins. Prostaglandins is another molecule, another type of fat that you don't discuss. It's not like it's saturated, unsaturated. It's just a different class of fats. So it's like a signaling fat. It's used as kind of like a flag. And it's primarily made up of fat. So these prostaglandins come in many shapes and sizes. And omega-6s, if they're used by this COX enzymes, the COX enzyme, if you want to look it up, then that enzyme will convert it to a different type of prostaglandin. So this prostaglandin molecule will then have detrimental effects in terms of increasing cancer growth. For the mechanism on that, please watch the other video where I go into the details of, or some of the details on how prostaglandins have that effect. Now, that said, consuming more omega-3 fatty acids seems to have the opposite effect. It turns into a different type of prostaglandin, which, as far as I understood it, converts it to another type of prostaglandin, which does not have this effect. Now, granted, let me give you a few numbers here. The Western diet has a ratio of about 15 to 1 omega-6s to omega-3s. And what was typically considered healthy, at least in just a general health context, but certainly in a cancer context, is having a ratio of 1 to 1. Huge difference there. Huge difference. So something for you to mull over. So that said, I suppose there are two things you can do. One, you can either try to reduce your omega-6 fatty acid intake, which from your like regular foods and whatnot, or you can try to increase your omega-3 fatty acid intake. But for you to consume enough to regulate your omega-6s with your omega-3s, that's a massive increase. So probably you'd want to reduce your omega-6 fatty acids. But that's just something to keep in mind. Also, on top of that, if you overconsume omega-3 fatty acids, it has the direct opposite effect of omega-6s in that it reduces inflammation, great, but then eventually it gets to a point where it becomes immunosuppressive. So reducing inflammation is immunosuppressive, but that's the point. That's what inflammation is, although a lot of people don't understand that. It's kind of one of my pet peeves. People don't understand that. Inflammation is your immune system. But I digress. The point is that if you overconsume omega-3s, meaning you consume a lot, more than you're supposed to, then it will have detrimental effects on your immune system. And you do want your immune system to be around to fight off things like cancer. Okay, so that was a lot of talking. Let me go ahead and finish this up by giving you a little rundown on everything that we just talked about. So, in conclusion, this is, as always, I always love to shame the people that loves to skip to the conclusions on these long videos, but I also understand people just want the answer. Unfortunately, that's not how I operate. But if you're skipping to this, I hear you. But if you have gone through the entire video, then as always, highly commendable, because I know it can be kind of heavy. But in conclusion, let's do this. This is not all encompassing, but because certain cancers rely on particular fat enzymes, so more heavily than regular tissues, which we talked about with like fatty acid synthase or CPT1 or those kinds, drugs can target those enzymes and do more harm to the cancer than regular tissues. Now, the exception to that was when the drugs were affecting CPT1, as I explained, but it did not have as many negative effects side effects when inhibiting the ATP citrate lyase. However, nutritionally, what we can do is also have an effect through ketogenic diet, and I explained some of the positives of that. Not that this applies to all cancers, and by fasting, so normal fasting through AMPK activation. Now, I also talked about how AMPK activation was not necessarily always a good thing, depending on the cancer type. And this is such a crucial point. That's why I wrote it here again. This is not universal to all cancers. Depending on cancer type, you would need to characterize the cancer, then figure out what kind of strategy would work based on its enzymatic profile, its epigenetic profile, its behavior, where it's located. All those different things have an impact. So distinguishing the type of cancer is critical. Okay, folks, that's what I've got for you. Thanks for slugging it out with me. Hopefully you learned something. Hopefully it was informative for you. There's so much more information out there than what I'm covering in this. This is just one review. They have more information, but it gets way into the weeds. And at a certain point, it's just like, what's the return on investment? If I spend an hour looking at one mechanism, is that really going to help us understand this? And the answer is no, not typically. So I still gave a decent amount of depth. But if you want to check it out, then I have my notes, so you can read it for yourself. And if you're interested in AMPK and fasting, the effect that fasting has on AMPK, I would recommend the next video, because that is going to be a contentious subject, in that certain individuals don't see as much of a benefit from fasting on the levels of AMPK and what implications that might have on cancer progression, as we discussed in this video. All right, so with that, I'll hope to speak with you in the future, and thanks for stopping by. Bye.